Hello humans, welcome back to Your Thoughts Are Valid, and I hope you all are doing great. So in today's video, we are going to learn about action potential. So this is a very important concept, although it's not compulsory that every year one question will come from this topic. From brain, it will definitely come, but from action potential, it's not sure. But even if any questions come from this concept, it's very much likely that you can make a mistake because this is a bit tricky to understand. So that's why we are going to learn about action potential in detail today. We are going to watch other videos as well, so that some videos where you can actually see how this process works, so that you can understand what action potential really is. So let's start with the video. Information flow through neurons. Okay, so they are showing that how information is flowing through a neuron. So it starts from dendrites. Okay, so dendrites actually collect electrical signals. So before you watch this video, I highly recommend that you must be aware with the structure of neuron. If you are not aware, or if you are not if you cannot memorize right now, so please watch the video on neuron first, only then watch this video. Okay, so yeah, dendrites collect electrical signals. Okay, dendrites collect electrical signals and they pass it on to cell body. So cell body integrates incoming signals from dendrites and generates outgoing signal to axon. Okay, integrate incoming signal from dendrite and generate outgoing signal to axon. Then what does axon do? Axon passes electrical signals to dendrites of another cell. Okay, another cell. Just remember that axon will pass these electrical signals to dendrites of another cell, or we can say that to the cells, or to an effector cell. Okay, so let's move on. So this is like the resting potential stage of a neuron. Okay, so here you can see that potassium ions are denoted by squares, blue squares, while sodium ions are shown by purple circles. So you can see that sodium ions are more on the outside of the cell. So the portion which is dark blue, that is outside of the cell, and the portion which is green, it is inside, that is the axon. Okay, so that is the axon, and the blue part is outside the axon. Okay, so that is outside cell blue part and that is inside cell green part. So yes, sodium ions are more on the outside, potassium ions are more on the inside. So what is polarization? Let's understand these two terms, polarization and resting potential, which I used when I showed you this picture. So let's see what is polarization. Polarization refers to the separation of electric charge across a cell membrane. Polarization refers to the separation of electric charge across a cell membrane. In neurons, polarization commonly refers to the resting membrane potential. Okay, when we are talking about polarization, we are referring to the resting membrane potential. Like when there is no signal, when your body is just at rest, there are no upcoming signals. Uh, so here the inside of the cell is negatively charged. Why it is negatively charged? Because potassium ions are attached to more positive ions, more positive molecules inside the cell only. That's why, sorry, more negatively charged molecules. Okay, potassium ions which are inside the cell, like the number of potassium ions inside the cell in resting potential stage is uh, more. So potassium ions are attached to the molecules which are more negatively charged than those molecules which sodium ions are attached to. So that's why inside of the cell is negatively charged relative to the outside. Okay, negative, there is more inside, there is more negative charge. Just remember the part that inside of the cell is more negatively charged in the state of resting potential. So this charge separation is maintained by the differential distribution of ions and the selective permeability of the membrane. So this whole pink part is the membrane. Okay, so here they are saying that this charge separation is maintained by differential distribution of ions. That is, inside there is more negative charge, outside it's negative, but relatively it's less, and the selective permeability of the membrane. What is resting potential? So resting potential is the membrane potential of a cell when it is not actively transmitting signals. So it is the cell, it is the it is the voltage. Just understand potential, resting potential as voltage. Okay, so they are saying, suppose you are at rest, there are no signals which are transmitting in your body. So at that time the resting potential of neuron or a cell would be minus 70 millivolt, minus 70 millivolt, with the inside of the cell being more negative than the outside. We already studied about this. This potential is maintained by the selective permeability of the cell membrane to different ions and the activity of ion pumps like the sodium potassium pump. So by selective permeability of cell membrane, they mean that cell allows few ions inside, few ions just outside. So it is selective, it is selecting which ions to send outside and when, which ions to let come inside of the cell and when. And then there are ion pumps, especially sodium potassium pump is really important when we are studying this concept of action potential. So this is about polarization and resting potential. So I hope it's clear till here that what is polarization and resting potential. So these two terms are used when your body is at rest, when there are no signals which need to be transmitted. Okay. So I hope it is clear till here. If you have any doubt, please do ask in the comment section. So now we will study about ion channels. But before that, I will understand from this slide after we watch a video, because you really need to understand the concept clearly. For that, I will show you one video. In that video, the person is explaining with, animation. So it's quite easy to understand what action potential really is. So let's watch the video.
What if everything you did, and thought, and felt could be communicated by pushing a button? It'd be like using the world's simplest app, one that just sends out a little ping, always at the same volume and length, to communicate everything from "it sure is cold in here" to "I love churros" to "boy, I sure would like to breathe sometime soon." Well, that is actually exactly how your neurons send all the impulses responsible for every one of your actions, thoughts, and emotions. When a neuron is stimulated enough, it fires an electrical impulse that zips down its axon to its neighboring neurons. But they've only got one signal that they can send, and it only transmits at one uniform strength and speed. What they can vary is the frequency or number of pulses, like this is distinct from this. And your brain can translate these signals, reading them like binary code, organizing them by location, sensation, magnitude, and importance, so that you know the difference between "turn up the thermostat" and "oh my gosh, I'm on fire." That buzz, that nerve impulse, is called the action potential. It's one of the most fundamental aspects of anatomy and physiology, and really life in general. It's happening inside of you right now, and we want to make sure that you understand what all that buzz is about. Before we delve into how neurons communicate, we gotta first understand a little bit of our old friend electricity. Basically, Think of your body as a sack of batteries. No, I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positively or negatively charged than others. And because opposite charges attract, we need barriers or membranes to keep positive and negative charges separate until we're ready to use the energy that their attraction creates. In other words, we keep them separated to build potential. A battery, just sitting on its own, has both a positive and a negative end, and the potential to release energy, but it doesn't do anything until it's hooked up to a flashlight or a phone or a kid's toy that lets those charges move toward each other, on the way converting electricity into light or sound or children's laughter. In much the same way, each neuron in your body is like its own little battery with its own separated charges. It just needs an event to trigger the action that brings those charges together. If you're thinking that this sounds more like engineering than anatomy, that might not be a bad thing. It might even help to think of your neurons in the same terms an electrician might use. Voltage, for example, is the measure of potential energy generated by separated charges. It's measured in volts, but in the case of your body, we use millivolts because it's a pretty small amount. In a cell, we refer to this difference in charge as the membrane potential. The bigger the difference between the positive and negative areas, the higher the voltage and the larger the potential. And just like there's voltage in your body, there's also current, the flow of electricity from one point to another. The amount of charge in a current is related both to its voltage and its resistance. Resistance is just whatever's getting in the way of the current. Something with a high resistance is an insulator, like plastic, and something with a low resistance is a conductor, like metal. Now, when we talk about these concepts in terms of you, we're typically talking about how currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membranes. And again, these membranes separate the charges, so they're what provide the potential to convert the electricity into something useful. Okay, now that we got Electricity 101 down, let's see how it works inside your nervous system. A resting neuron is like a battery just sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential, and it sits at around negative 70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. This creates a difference in the concentration of sodium and potassium, and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient, and you probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around negative 55 millivolts. But some others are ligand-gated channels. They only open up when a specific neurotransmitter like serotonin or a hormone latches onto it. Then we also have mechanically gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions. This movement of ions is the key to all electrical events in neurons, and thus is the force behind every single thing we think, do, and feel. Of course, not all of your body's electrical responses are the same, and neither are the flows of ions going in and out of your neurons. If only a few channels open and only a bit of sodium enters the cell, that causes just a little change in the membrane potential in a localized part of the cell. This is called a graded potential. But in order to send long-distance signals all the way along an axon, you need a bigger change, one big enough to trigger those voltage-gated channels. That is an action potential. And your best bet for making that happen is to depolarize that resting neuron. I mean, cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It all starts with your neuron sitting there at resting state. All of the ion channels are closed and the inner voltage is resting at negative 70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now, the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about negative 55 millivolts. Remember that number, because this is an all-or-nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. But kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, you're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about positive 40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them. And that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to negative 75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level. Now, when part of an axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter how strong. This is called the refractory period, and it's there to help prevent signals from traveling in both directions down the axon at once. So that is the surprisingly simple app that your nervous system uses to let you experience the world. And because the voltages in this process are always pretty much the same, the initial threshold around negative 55 millivolts and the peak depolarization at 40 millivolts, your neurons only communicate in a single monotone buzz. It doesn't matter if it's a spider on your knee or an elephant, a paper cut or a stab wound, the strength of that action potential is always the same. What does change is the frequency of the buzz. A weak stimulus tends to trigger less frequent action potentials, and that includes if the stimulus is coming from you, like your brain telling your muscles to perform some task. If I need to do something delicate, like pick up an egg, the signal is low frequency. But a more intense signal, like trying to crush a can, increases the frequency of those action potentials to tell your muscles to contract harder, and the message turns into something that you can't ignore. Action potentials also vary by speed or conduction velocity. They're fastest in pathways that govern things like reflexes, for example, but they're slower in places like your glands, guts, and blood vessels. And the factor that affects a neuron's transmission speed the most is whether there's a myelin sheath on its axon. Axons coated in insulating myelin conduct impulses faster than non-myelinated ones, partly because instead of just triggering one channel at a time in a chain reaction, a current can effectively leap from one gap in the myelin to the next. These little gaps are the delightfully named nodes of Ranvier, and this kind of propagation is known as saltatory conduction, from the Latin word for leaping. But what happens when an action potential hits the end of its axon and is ready to do more than leap and jump all the way to another neuron? That you will find out next time. Today, you learned how your body is kind of like a big bag of batteries, and how ion channels in your neuron
Okay, so for now, let's just watch this one and understand other concepts. Then we will come back and watch another video. Till then, let's understand. Okay, so first let's understand ion channels. Okay, so ion channels are specialized protein structures which are embedded in cell membranes that allow specific ions to pass through the membrane. These channels are essential for controlling the flow of ions in and out of cells. Channels can either be gated, that is controlled by various factors, or always open. They are responsible for generating electrical signals such as action potential by regulating the movement of ions across the cell membrane. Uh, okay, so here we are shown some ion channels. Okay, so here is voltage gated sodium channel. So it is generated by, difference in charges. So that's why we are saying that voltage gated it is. Then there are voltage gated potassium channel, and there are mechanical gated ion channels. Okay, sodium channel, potassium channel, and ion channel. So what is sodium potassium pump, or semi-permeable? These are semi-permeable. So let's understand this. So a semi-permeable membrane is one that allows certain substances to pass through while blocking others. Okay, so they just allow few substances to pass, and while blocking uh, others by while blocking other substances. So consider it as sieve. Okay, so it's like, sieve just allow the liquid part, and don't allow the solid part. So like that, consider semi-permeable membrane as a sieve. Sodium potassium pump is a critical protein complex found in cell membranes, especially in neurons. It actively transports sodium ion out of the cell and potassium ion into the cell. Okay, sodium ion out of the cell and potassium ion into the cell against their respective concentration gradient. And this process requires energy in the form of ATP and helps maintain resting membrane potential and proper ion balance. Help maintain resting membrane potential and proper ion balance. Then what is selective permeability? Voltage gated channels. Okay, so selective permeability refers to the property of membrane that allows only specific ions to pass through. Okay, property of membrane that allows only specific ions to pass through. Voltage gated channels are a type of ion channel that opens or closes in response to changes in the membrane potential or voltage across the cell membrane. These channels are critical for generating action potential and other signals. Examples include voltage gated sodium channels, and voltage gated potassium channels. So here we learned about sodium potassium pump and voltage gated channels. So I hope it's clear till here, because until and unless you understand what are these channels, you won't be able to understand how action potential is generated. So there is also one thing which I would like to tell you, that what is the difference between semi-permeable and selectively permeable. So semi-permeable membrane permit the movement of solvent molecules through them, but prevent the movement of solute particles. Okay, while in selectively permeable membrane, it's like normally semi-permeable, but it allows selective passage of solutes through them. Okay, selective permeable membrane is also semi-permeable. It's just that these allow only specific ions to pass through them. Okay, so I hope the difference is also clear. So what is ATP? ATP stands for adenosine triphosphate. It is a molecule that serves as primary energy currency in cells. It is high energy molecule that stores and transfers energy within living organism. It plays a crucial role in various cellular processes, including metabolism, muscle contraction, active transport, and signal transduction. Okay, so it's not that important to remember. It's just that I wanted to explain what ATP is when I mentioned it. Okay, so before we learn about depolarization, repolarization, let's look at this. Okay, so yeah, uh, inside there is net negative charge. Here we are talking about resting potential itself, as we learned that there are more negative charge, that negative charge is more on the inside of the cell. And as you can see on the outside of the cell, it's more it's positive, and on the inside it's negative. So as I told you, what is the resting potential of a membrane, of a cell membrane or of a cell? It's minus 70 millivolt. Minus 70 millivolt. Okay. So just understand how these gates work. So as we saw, here we will be talking about sodium channel. Okay, voltage gated sodium channel. So you can see when it's open, it looks like this. Okay, when it's closed, it's look like this, and when it's inactivated, it's look like this. Okay, so you should remember this. Okay, then moving on, threshold. Threshold is the membrane potential level that must be reached to initiate an action potential. Okay, so threshold is membrane potential level that must be reached to initiate an action potential. Once the threshold is surpassed, voltage gated sodium channels open, allowing a rapid influx of sodium ions and triggering the depolarizing phase of an action potential. The threshold is typically around minus 55 to minus 50 millivolt in neurons. Okay, so understand it like, suppose someone is sleeping. So initially you will just try to tap that person, tap any part of that person, slowly, very slowly, to wake that person up. But when you see that it's not happening, that the person is not waking up, you tap, uh, with bit with a firm hand. Okay, like you will start tapping with more force. Okay, so that you can wake that person up, or like just take the name of that person in a higher voice. Okay, so that you can wake that person up. So just like that, there are many signals. There are many signals which our brain can process, but it's just that uh, like for an action potential to generate, there must be a particular threshold to reach. Otherwise, it will get very difficult for our body to you know to experience all those sensations or pass on those messages, basically transmit signals. So there must be a particular threshold which is necessary so that an action potential can be generated. Like the stimulus must be effective enough so for an action potential to be generated. Once this threshold is surpassed, voltage gated sodium channels open. Okay, so once you reach that threshold, okay, now what happens? Voltage gated sodium channels will open. So what will happen when these channels open? So there is a rapid influx of sodium ions, like many sodium ions come inside the cell, and this triggers the depolarizing phase of action potential. And the threshold in neurons is typically minus 55 volt. Okay, so whenever you will be asked about threshold in neurons, it's minus 55 millivolt. What is depolarization? So depolarization is a change in the cell membrane potential, that is voltage, where the inside of the cell becomes less negative or even positive compared to the resting potential. Okay, so in depolarization, what happens? That inside of the cell becomes positive. We will use the term positive because it's like outside it's negative now and inside it's positive. In resting potential, inside it was negative and outside it was positive, but now it's vice versa. So the cell becomes negative, or we can less negative, or we can say positive compared to the resting potential, and this change is often caused by influx of positively charged ions. Okay, because sodium ions moved inside, there was a rapid influx of sodium ions inside the cell. That's why, it became positive. Depolarization is a key step in the generation of an action potential. Okay, if depolarization won't happen, action potential won't be generated. Then what is action potential? So action potential is a rapid and transient change in the membrane potential of a neuron or muscle cell. Okay, rapid and transient change in the membrane potential of a neuron or muscle cell. It is a brief electrical signal that travels down the length of the cell's membrane, transmitting information within the nervous system. And action potential is initiated when the membrane potential reaches a certain threshold, leading to depolarization, followed by repolarization and hyperpolarization. Okay, an action potential is initiated when membrane potential reaches a certain threshold, leading to depolarization. And after depolarization, what happens? Repolarization and hyperpolarization. So, let's just watch another video to understand this concept. Uh, just a minute. Yes. So in this graph, it is shown that, it's minus 55 millivolt. Threshold is minus 55 millivolt. Okay. Now let's understand action potential with the help of a video.
This is a neuron, which has four main parts. The dendrites receive information. The cell body processes and integrates that information. The axon carries the information along long distances from one part of the neuron to another, and the axon terminal transmits the information to the next cell in the chain. A bundle of axons traveling together is called a nerve. Nerves can be very long, as they often need to transmit information over long distances. As we just saw, the dendrites are the part of the neuron that receives incoming signals. Based on the strength of this incoming stimulation, the neuron must decide whether to pass that signal along or not. If the stimulation is strong enough, the signal is transmitted along the entire length of the axon in a phenomenon called an action potential. When this happens, we say the neuron fires. Transmission of a neuronal signal is entirely dependent on the movement of ions or charged particles. Various ions, including sodium, potassium, and chloride, are unequally distributed between the inside and the outside of the cell. The presence and movement of these ions is not only important when a neuron fires, but also at rest. To start, let's think about the positively charged sodium and potassium ions. When a neuron is not sending a signal, it is considered to be at rest. In a typical neuron, in its resting state, the concentration of sodium ions is higher outside the cell than inside. The relative concentration of potassium ions is the opposite, with more ions inside the cell than outside. This ionic separation occurs right at the cell membrane and creates a chemical gradient across the membrane. Because ions are charged particles, we also need to consider their charge when thinking about their distribution across the membrane. At rest, there are more positively charged ions outside the cell relative to the inside. This creates a difference in charge across the membrane, which is called an electrical gradient. Together with the chemical gradient we already mentioned, we refer to this ionic imbalance as the electrochemical gradient. The difference in total charge inside and outside of the cell is called the membrane potential. At rest, when no signals are being transmitted, a neuronal membrane has a resting potential of approximately minus 70 millivolts. This means that the inside of the cell is approximately 70 millivolts less positive than the outside. Both the chemical and electrical gradients we just discussed contribute to establishing this potential. While the inside of the cell has a net negative charge and the outside of the cell has a net positive charge, the charges line up at the membrane and the bulk solution on either side is actually electrically neutral. The resting membrane potential is the point where the cell has achieved electrochemical equilibrium. This means that the concentration gradient and the electron gradient for each ion is equal and opposite. Ions cannot simply move across the membrane at will. Instead, they need a protein embedded in the membrane to facilitate their movement. Most ions cross the membrane through a structure called an ion channel. Ions move through channels by passive diffusion along their concentration gradient. Some ion channels are always open, but many require a signal to tell them to open or close. For example, voltage-gated channels only open when the membrane potential reaches a certain value. On the other hand, ligand-gated ion channels are triggered to open when they are bound by a specific molecule. Mechanically gated ion channels open in response to physical forces, such as changes in length or changes in pressure. Most ion channels are selectively permeable, meaning that they only allow one or a small subset of ions to pass through. Voltage-gated ion channels, for example, typically only allow a single ion to cross the membrane when they open. This means that we need separate channels for each ion, i.e., voltage-gated sodium channels, as well as voltage-gated potassium channels. As ions move through a channel and cross from one side of the cell membrane to the other, they cause the membrane potential of the cell to move away from its resting potential. If the resulting change in membrane potential is small, we call this a graded potential. Graded potentials can vary in size, can be either positive or negative, are transient, and typically do not result from the opening of voltage-gated ion channels. When ion channels open and a graded potential occurs, the neuron moves quickly to reset its membrane potential to resting values. This is accomplished primarily by the use of the sodium-potassium pump, which uses the energy generated by ATP hydrolysis to actively transport ions across the membrane against their concentration gradient. In other words, sodium is transported to the outside of the cell, where its concentration is higher, and potassium is transported back into the cell, where its concentration is higher. One cycle of this pump transports 3 sodium ions outside the cell and brings 2 potassium ions inside the cell. This unbalanced charge transfer contributes to the separation of charge across the membrane and also to the ionic concentrations we see at rest, thus restoring the chemical and electrical gradients to their resting levels. Maintaining these ionic balance in neurons is so important that this process can account for 20 to 40% of the brain's total energy use. Only when the resting membrane potential and ion distributions are maintained at precise levels will the neuron be poised and ready to fire an action potential. When the outside stimulation is large enough to bring the membrane potential in the neuron body up from -70 millivolts to the threshold voltage of -55 millivolts or higher, this triggers an action potential at the axon hillock, which then travels down the axon. Voltage-gated sodium channels have 3 states: open, closed, and inactivated. At rest, the sodium channel is closed. Once the cell membrane reaches the threshold voltage, the channel changes to an open position, and sodium rushes into the cell because of the electrochemical gradient. As positive sodium ions enter the cell, the membrane potential becomes less negative and more positive as it approaches 0 millivolts. This is called depolarization. Eventually, the voltage gradient goes to 0 and beyond 0, up to a positive 30 millivolts. This is called an overshoot. As the membrane potential becomes positive, the sodium channel inactivation gate shuts, making the channel inactivated. This stops the flow of sodium ions into the cell. The change in membrane potential also opens the voltage-gated potassium channels, though they open and close more slowly. Because of the potassium electrochemical gradient, potassium ions flow out of the cell, making it less positive and eventually negative. This process is called repolarization. Because the potassium channels are a little slow to close, for a brief period the membrane potential is hyperpolarized. It's more negative than the resting potential. During hyperpolarization, the potassium channels close. Throughout all this, the sodium-potassium pump is still working. The pump restores the chemical gradients by putting the sodium and potassium back in place. And the pump reestablishes the potential gradient by moving more sodium ions out than potassium ions in. This returns the membrane potential back to its resting potential. During repolarization, the inactivated sodium channels won't respond to any stimulus at all. During this time, the neuron is in its absolute refractory period, the period of time when a nerve cannot fire another action potential no matter how strongly it's stimulated. The absolute refractory period prevents action potentials from happening again too quickly and prevents the action potential from traveling backwards along the axon. During hyperpolarization, the sodium channels are closed and the inactivation gate opens. There is no change in sodium flow, but now they could be opened again. This is called the relative refractory period, because while the sodium channels could open, it would take a larger than usual stimulus to reach threshold, because the cell is hyperpolarized due to the potassium still leaving the cell. The amplitude of the action potential for a particular neuron, that is, the maximum voltage in one neuron during an action potential, never changes. An action potential doesn't get bigger with a bigger stimulus. It's all or nothing. It either happens or it doesn't happen. What can change is the frequency of the action potential. A neuron might fire many more times per second in response to, say, an intense pain, and less frequently in response to a gentle breeze. Some axons transmit action potentials faster than others. One variable that increases conduction velocity is the presence of myelin sheaths around axons. Myelin speeds up transmission through a process called saltatory conduction, in which the action potential signal appears to jump along the part of the axon covered by the sheath. In the peripheral nervous system, the sheaths are formed from glial cells known as Schwann cells. There are small gaps between Schwann cells called the nodes of Ranvier. The action potential appears to jump from node to node, speeding the transmission. In the central nervous system, the sheaths are made by cells known as oligodendrocytes. To review, with no stimulus, the membrane is at its resting potential. A small stimulus causes a graded potential, and a stimulus above the threshold creates an action potential and the neuron fires.
Okay, so I hope it's clear now. Let's move back to the slides. Okay, so I think threshold, depolarization, action potential, all is clear to you. Let's move on. Okay, so here it's showing that, the signal has crossed the threshold. So now the potassium channels open and there is sodium influx. So that is shown in this picture. Then when depolarization happens, it is shown here, like when depolarization, how the cell looks like, how the cell membrane looks like. Also, the sodium channels get inactivated, okay, not closed, just inactivated. And this is potassium channel, so this gets open, okay, in depolarization. Then what is repolarization? Repolarization is the process that follows depolarization during an action potential. It involves the restoration of the cell's membrane potential to its resting state after depolarization. This is achieved by opening voltage-gated potassium channels, allowing potassium ions to leave the cell and bringing the membrane potential back to its negative resting value. Hyperpolarization is a change in the membrane potential where the inside of the cell becomes more negative than its resting potential. This often occurs after an action potential when the cell's membrane potential overshoots the resting potential due to the continued activity of potassium channels or the closure of certain ion channels. Hyperpolarization makes it more difficult for the cell to reach the threshold for initiating another action potential. Okay, so here they have shown repolarization process. Okay, sodium channels are still inactivated and potassium channels are open. Then what is refractory period? Refractory period is a brief period of time after the initiation of an action potential during which the cell is less responsive to a new stimuli. This period is crucial for maintaining proper sequence and timing of action potentials and prevent the cell from generating excessively rapid or continuous firing. What is absolute refractory period? It is the early phase of refractory period during which neuron is completely unresponsive to any new stimulus, no matter how strong. This period coincides with the depolarization and repolarization phases of action potential and is due to the inactivation of voltage-gated sodium channels. Relative refractory period. The relative refractory period follows the absolute refractory period and is a time during which the neuron can be stimulated again, but only by a stronger than normal stimulus. This period is characterized by the continued hyperpolarization of the cell membrane, which makes it more difficult for the membrane potential to reach the threshold for initiating a new action potential. So I hope these three periods are clear to you. It's already explained in that video, so I hope it's not tough for you to understand now. Then here they have shown absolute refractory period. How does it look when absolute refractory period is going on? This is relative refractory period. Okay, so here, this happens in the repolarization process. This happens when the cell has reached hyperpolarization state. Then what is all or none phenomena? It is very important to understand, although you have already understood in that video, let's just revise what all or none phenomena is. So it refers to the fact that an action potential either occurs fully or doesn't occur at all in response to a stimulus. If the stimulus is above the threshold, a full action potential is generated. If it is below the threshold, no action potential is produced. The strength of a stimulus does not result in a larger or stronger action potential. It only affects whether the action potential occurs or not. So this graph is very important. You must remember this graph. Okay, so as you can see here, there is one stimulus. Okay, so when the stimulus reaches the threshold, so in the resting potential, the voltage is minus 70 millivolt. When it reaches the threshold, that is minus 55 millivolt, depolarization starts. Okay, so sodium ions start getting in. That is when action potential is generated. Then the third one is repolarization period. Okay, so what happens in repolarization? Potassium ions moves out. Okay, what happens in repolarization? Potassium ions move out. Then hyperpolarization occurs, which is the fourth state, and then again resting state. Okay, that is the fifth state. Okay, resting state is again what is minus 70 millivolt. Threshold is minus 55 millivolt. And what is the action, like what is the peak of depolarization? what is the voltage at the peak of depolarization is plus 40 millivolt. Remember this one, okay? Plus 40 millivolt. Then there are few more concepts which you need to understand, that long-term potentiation. So it is a persistent and strengthening increase in synaptic strength between two neurons that result from repeated and intense stimulation. Okay, so how it works, how LTP works. So there is synaptic activation. LTP occurs at synapses, which are connection between neurons where communication takes place. When a presynaptic neuron, that is sending neuron, repeatedly and strongly activates a postsynaptic neuron, which is receiving neuron, it triggers the change, it triggers changes in the strength of the synapse. Okay, then what is a strengthening of synaptic transmission? During intense and repetitive stimulation, postsynaptic neuron becomes more responsive to the signals coming through the presynaptic neuron. This is achieved through changes in the efficiency of neurotransmitter release, receptor sensitivity, and alterations in the density and function of ion channels in the postsynaptic membrane. So this is what? This is long-term potentiation. Okay, it is just about persistent and strengthening increase in synaptic strength between two neurons, okay, which occurs from repeated intense stimulation. There are two processes which are involved in LTP: synaptic activation and strengthening of synaptic transmission. So what happens in synaptic activation? Connections between neurons where communication takes place. A synapse occur, LTP occurs at synapses, and you already know what synapses are. Okay, so when a presynaptic neuron, that is sending neuron, repeatedly and strongly activates postsynaptic neuron, it triggers change in the strength of synapse. Okay, and during intense and repetitive stimulation, postsynaptic neurons becomes more responsive to the signals coming from the presynaptic neuron. I hope this much is clear to you. Then structural changes. In some cases, LTP can lead to structural changes at the synapse. This might involve an increase in the number of synaptic receptors or the growth of new dendritic spines, which are tiny protrusions on the dendrites of neurons that receive signals from other neurons. These structural changes can contribute to the long-lasting nature of LTP. Enhanced communication. Net result of LTP is that the communication between presynaptic and postsynaptic neuron become more efficient. When presynaptic neuron releases neurotransmitters, postsynaptic neuron is most likely to generate an action potential, leading to a stronger and more effective transmission of the signal. So yeah, that is it for today's video, and I hope action potential is clear to you all now. And yes, I will provide the link in the comment, in the description below, for the two videos we watched today to understand this concept. So yeah, that is it for today's video. If you still have any doubt, please do ask in the comment section, and thank you so much for watching.